Hi, welcome everyone, and sorry that I am just a minute or so late. So this afternoon I am planning to go through the paper 2 of the OCRA 2018 paper. Just need to be aware that I am not going to manage to go through every question this afternoon. I'm going to get up to question 20 out of the 22 that is on the paper. I just haven't had time today to quite finish it off, which I would have loved to have done. So what I'm going to do is add an extra video on. I'm just going to kind of do it and add it on so that this going through this paper will actually be cut across two live videos in effect. So you will be able to access them all, but not immediately. So let's have a look at this paper. And starting with the section A, which is the multiple choice sections. Oh, just to say that I will be keeping an eye on any chat that comes up. I will be glancing across at my second screen. So if about the answers that I'm giving and why they are the answers that they are, do tap away. Just be aware that I am running about 10 seconds ahead of what you are hearing. So there might be a little bit of a delay and I might have to jump back. But let us just jump in now with section A and with the multiple choice part of the paper. So which of the following best describes a microscope with high resolution? A, microscope can distinguish structures that are very close together. I'm loving that answer, but I'm going to check the others. The microscope can view structures that are very small. That is about magnification, not resolution. The microscope is capable of high magnification. Again, magnification, not resolution. And the microscope has an inbuilt eyepiece graticule. Absolutely irrelevant. So it is A. The image below shows a tardigrade, the most bizarre organisms. They talk about extremophiles. These things can survive anything. High temperatures, high salinity, radioactivity, the lot. Anyway, moving on. So, viewed from underneath, here its little legs, and the magnification is times 110. How long is the tardigrade in real life? This requires you to have the paper in front of you at an appropriate magnification. You need the paper at the actual size. So what I did was that I went back to, and I put it up on the screen from the website, and I put it to 100%. And I then measured from one end to the other, and it worked out at 87 millimeters. I know that magnification equals image, which is the picture, over the actual, which is the actual size of the real thing. So rearranging this equation gives me the actual size as image over magnification. I got it as 87 millimeters, and therefore I ended up with a value of 0.79 millimeters. And then I looked at all the answers. And suddenly I'm realizing that it's hardly about the calculation. It's actually about understanding all of these different units and what they mean. So I'm kind of going, well, the numbers that's closest is C, but I need to make sure that this is right. So 0.79 millimeters does end up being 7.9 times 10 to the minus 4 meters. And so I plumped for C, and I'll show you that in a minute. But let me first just clarify all this unit stuff. 1 meter, as we all know, is made up of 100 centimeters, but each of those centimeters is 10 millimeters. And that means 1 meter is made up of 1,000 millimeters. 1 millimeter, so one of those little tiny divisions in your ruler, can be split into another 1,000 divisions. And each one of those divisions is a micrometer. So 1 millimeter equals 1,000 micrometers. Your head might be starting to explode now. But then really start thinking about One of those tiny micrometers, a thousandth of a millimeter, and that can be split into another 1,000 parts, which is the nanometer. So 1 micrometer is made up of 1,000 nanometers. My general advice to you is never use centimeters in biology. Always switch any centimeter length that you measure into millimeters. And it's worth noting that if we're writing this in standard form, that a millimeter is 1 times 10 to the negative 3 of a meter. It's 1,000th of a meter. And a micrometer is a millionth of a meter, 1 times 10 to the negative 6. So, and a nanometer is 1 times 10 to the negative 9 meters. Hopefully that helps a little bit unwrap this question, but I've kind of worked out that I'm going to plump for C, given that I haven't got the actual paper in front of me, and that was the answer. So here we have a graph of an enzyme reaction and showing the amount of volume, the volume of oxygen collected over a period of time. So it's obviously going to go up fairly rapidly, but the gradient, the rate of production of oxygen is going to decrease. And finally, the volume of oxygen is going to reach the maximum. This gradient ends up being 0, no more oxygen being produced. Which of the following shows the rate of reaction at 30 centimeters? They've emboldened at. And that's because they're wanting you to work out the rate of reaction at 30 centimeters at that particular point in time, not over the first 30 centimeters. It's a very different calculation. If you're asked to work out the rate of reaction at a particular time, and time is obviously on the x-axis, this requires you to draw a tangent to the line. That is, it's a straight line that kind of kisses the line just at the point you're being asked for, in this case, 30 seconds. And I haven't got this perfectly right because it's actually really quite tricky to do. But what you're trying to do is get the angle here and the angle here to be exactly the same, because then you're relatively sure that you've got the angle of the line correct as it just touches the point that you're interested in. And then, of course, you work out, because the rate is the gradient of the line, you are now working out the gradient of the line at this point in time. And that, of course, is, and if we're talking about rate, remember that you've got to divide something by the time. And so it is the volume of oxygen produced over a particular time. Now, because this line is a straight line, the gradient is the same across any part of it. So I have totally arbitrarily chosen 10 seconds. And can I just point out that this is not done particularly accurately either. I can immediately see that these two lines aren't perfectly parallel. So this isn't going to be a superbly accurate answer, but I'm kind of going, okay, there's an 8 centimeter cube difference in oxygen over 10 seconds. So 8 over 10 equals 0.8 centimeters cubed per second. And as you can see, that is closest to answer A. And so I would have gone with answer A. And I got it right. Now, it's worth noting that if you had calculated the average rate over the first 30 seconds, in other words, it would have in effect been the straight line from 0, to 30, you would have got 1.5. You would have ended up plumping for D. But that was the rate over the first 30 seconds, not at 30 seconds, which requires the drawing of a tangent. Okay, which of the following processes involves the formation of ester bonds? Synthesis of polynucleotides. Yeah, that is true because it forms phosphodiester bonds. And those phosphodiester bonds are basically two ester bonds, diester, on either side of the phosphate group. One side connecting with the sugar molecule in the nucleotide above, and one connecting with the sugar molecule in the nucleotide below. That's why they're called phosphodiester bonds. So, definitely one. Two, absolutely, ester bonds are in triglyceride between the carboxylic acid group on the fatty acid and the OH group on the glycerol. That's where an ester bond is formed. And it is not in polypeptide because that is a peptide bond. So only 1 and 2, that gives me the answer B, which was the correct answer. Which of the following could not be, sorry, just smiling at a comment. Thank you, Mr. Ibrahim Samir, thank you. Which of the following could not be an amino acid? So here is my standard drawing of an amino acid. Notice that you need an amine group on one side and a carboxylic acid group on the other side. It's why it's called an amino acid. So I'm looking for an amine group and a carboxylic acid group. The great big groups that you don't recognize are very likely to be the R group. So A, I've got my amino group or amine group here. Here's your central carbon. The skeletal formula doesn't show you every hydrogen, so there would have been a hydrogen here because every carbon has to have 4 bonds, 1, 2, 3, 4. But we have got a carboxylic acid group here, and here is the R group. B, here's my amine group, here's my central carbon here, I'd have a hydrogen here, here's my carboxylic acid group, here is my R group. So B is an amino acid. C, I've got my amine group, but I've got no carboxylic acid group. So I'm not liking the look of C. And finally, D, here's my amine group, here's the central carbon, I'd have my hydrogen here, here's my R group, and there is the carboxylic acid group. So it is C. Tricky question, I think. So now we've got to show the correct sequence of missing words. So I'm going to start reading. Lipids are a diverse group of chemicals that are neither polar nor charged, yeah, okay, and hence are insoluble in water. Yeah. The something nature of the heads of the phospholipids, well, that is hydrophilic because phospholipids have hydrophilic heads. So that reduces me down to A or B. It isn't, oh, sorry, I've just realised, and D. It certainly isn't C. That allows them to form membranes. Something also contain fatty acids and form part of the membrane. So it's not triglycerides because triglycerides do not form part of the membrane. Triglycerides are your fats and oils that are in your kitchen cupboards, in your foods. They do not form part of the membrane. So I'm now taking out B. I'm now down to A and D. And there's something that something also contain fatty acids and form part of the membrane. I know cholesterol molecules form part of the membrane. I don't think they have fatty acids in them. And I'm kind of thinking glycolipids do, because glycolipids are just lipids with some carbohydrate on them. So I'm veering towards A at the moment. Lipids can also be used for energy in the store of triglycerides. Yeah, I'm still thinking A, definitely B's out. So I'm still thinking either A or D. Some hormones are also lipids, and they are similar in structure to, now I know that is cholesterol molecules. because your hormones like testosterone and estrogen are actually steroid hormones, and cholesterol is a steroid. So I know that it is A, because we've got, and it absolutely fits in with my hunch that glycolipids, of course, they're going to have fatty acids in them. Cholesterol molecules do. Cholesterol molecules are steroids, like some of those steroid hormones. Which of the following statements about antibiotic resistance is correct? A, right, we're looking for the correct statement. All antibiotics cause mutations. Well, that's a load of rubbish. Antibiotics do not cause mutations in bacterial DNA. Mutations are caused totally randomly, and they would be their rates would be increased by things that are mutagenic. So things like X-rays, gamma rays, all those electromagnetic radiation, some chemicals, but not antibiotics. Antibiotics resistance in bacteria is evidence to support Darwin's theory of evolution by natural selection. Absolutely, I'm thinking that is correct. The development of antibiotic resistance in bacteria is an example of genetic drift. No, it isn't. It's an example of natural selection. Genetic drift is the taking out of alleles from a population due to a chance event, not due to natural selection, which of course selects those advantageous alleles. So this is not true. And I've got something here that isn't letting me, I've got a box that isn't letting me read all of this. Development of, this is the stabilizing selection option. It isn't stabilizing selection. Antibiotic resistance is an example of directional selection. So this is not true either. So it is B, and that was the right answer. Which of the following antibodies increase the phagocytosis of pathogens? Right, well, opsonins certainly do, because those are the ones that they grab hold of the antigen, and then the constant region of the antibody that is the opsonin links with the phagocyte, generally a neutrophil. So opsonins definitely. Agglutinins also do, because they link those are antibodies that can pull together two different antigens. So they create a mesh of two different pathogens. So they create a kind of mesh of pathogen and antibody, which increases phagocytosis. So that one. Antitoxins don't increase the phagocytosis of pathogens, because antitoxins, those are antibodies that bind with the toxin, thereby neutralizing them, because they stop the toxin binding to a receptor on a cell and bringing about the toxic effect. So it is only 1 and 2, so it's B, and that's correct. Which of the following describes an autoimmune disease? A disease in which an individual's own body cells are antigenic. That's right. That's right. Because really, let's face it, most of the markers on our cells, on our own cells, are not antigens. Antigens, by definition, are things that cause an immune response. We have markers on all of our cells that tell all of our immune cells that our cells belong to our body and therefore do not cause an immune response. The markers on our cells that declare our cells as self are not antigenic. However, in the situation of an autoimmune disease, those markers become antigenic because they cause an immune response. They cause our own immune system to attack our own body cells. So that is true. B, let me just go through the others though. A B, a disease in which a pathogen attacks cells of the immune system. That isn't what happens in autoimmune diseases. That happens in diseases like AIDS. but that isn't an autoimmune disease, it's a pathogenic disease. A disease that prevents the production of antibodies? No. And a disease in which an individual has gained immunity? Absolutely not. So the answer is A. During which stage of the cell cycle does semi-conservative DNA replication occur? It is the synthesis phase. It is the only phase in which it happens, so it is D. Cell cycle includes a number of checkpoints. Which of the following statements about the cell cycle is correct? If damaged DNA is detected at a checkpoint, apoptosis is triggered. Ooh, I quite like that answer. I'm not absolutely sure that at every checkpoint, apoptosis is triggered, but I'm kind of going, okay, let's keep on reading. If damaged DNA is detected in the G2 checkpoint, the cell cycle is halted and the cell tries to repair the damage. The G2 checkpoint occurs just before mitosis. And I know that that is true. The DNA has been, the extra copy of DNA has been synthesized, the DNA has been replicated. And so it makes sense that that DNA it tries to be repaired before mitosis occurs. So I am liking that response too. If a mistake is detected at a checkpoint, the cycle reverts to an earlier checkpoint and is repeated. No, that isn't true, because that would be saying that you'd be going from your G2 checkpoint back to your G1 checkpoint, and you'd be starting DNA synthesis all over again. Well, you'd end up with 4 copies of the DNA in the cell, not just 2. That doesn't happen. The G1 checkpoint checks for mistakes in DNA replication. That is also not true, because the G1 checkpoint occurs before the DNA replication occurs. So I've got to choose between A and B. And the bottom line is I am more sure of B than I am of A. So I would have plumped for B, and it was the right answer. It's worth noting that A was the other common mistake on that question. Okay, turtle doves, once common, numbers are in decline. Farmers can claim money if they encourage the survival. Which one of the following is relevant? Well, one isn't relevant because we're not talking about the trade of turtle doves, even though it sounds like they're an endangered species. So I don't think that one fits at all. The Rio Convention on Biological Diversity gives guidelines about how certain species, or how species can be, their populations can be increased. So I think that is an option. And the countryside stewardship scheme is absolutely an option, because that is something that encourages farmers to increase the biodiversity on their farms and to take action to do so. So it is only 2 and 3. So I'm thinking that it is C, and that did end up being the right answer. In which of the following processes do bacteria not play an active role? Well, this to me is a little bit of a gift of a question, because the first answer I am absolutely confident is not the case, and it is bioinformatics. Bioinformatics is basically the computer data processing of the stupid amounts of data that come out of genomic research into our genome, into all of our genes. So, and I'm just checking, yeah, bioremediation definitely uses bacteria, cheese making does, and so does manufacturing human insulin, because that is also biotechnology. So it is A. Excuse me. Okay, mycoprotein is a food produced using the fungus Fusarium venenatum. Which statement is mycoprotein correct? The production of protein is slower than in plants and animals. Well, that is not correct, because the industrial production of Fusarium actually produces protein at a rate much faster than in animals and much faster than in plants. So I'm absolutely confident that's not true. Production is dependent on seasons. Absolutely not true, because it's an industrial process, can happen at any time. Can use waste products as a substrate, in other words, as nutrients. I absolutely know that to be true. And there are no ethical issues associated with production. I wouldn't say, I can't think of any huge ethical issues with using a fungus for food. I'm aware that some people might choose not to eat something that is fungal, but then you wouldn't be eating mushrooms. And some people don't like eating things that have grown on, in effect, a waste product. You could say that can happen to most of our agricultural food, because manure is spread on the ground. And personally, I would say that is a far more offensive waste product than the substrates that are used to grow fusarium, because those are things like molasses, as in effect spare sugars from other industrial processes. So I can't think of, I'm absolutely sure it's C, but it could have been D. So, but I don't think it is. So it's C, and that was the right answer. Which of the following statements about ecosystems is not true? Right, so let's go through these. Ecosystem is affected by biotic and abiotic factors. That is true, therefore it's not true. An ecosystem is all of the organisms and habitats in a large area. It's not necessarily in a large area. It can be in a small area. You can have a small ecosystem. An ecosystem is dynamic. Absolutely, that is true. There is a flow of biomass between trophic levels in an ecosystem. Absolutely true, every time. So it's got to be B, even though it doesn't originally, when you first read it, you kind of go, oh, that sounds right. But actually, it's not always a large area. So it is that one. So 16, on the insert, we have onion cells undergoing mitosis. Name the stage of A. Well, that to me is, and particularly as these are in the right order, I absolutely know that this is metaphase. This is interphase. This is prophase because we've got the chromosomes visible. This is metaphase because the chromosomes have all gathered on the equator, and their centromeres are attached, are now all at the equator. So even though they look a little bit messy, they are all gathered on the equator. This is anaphase, this is telophase, and this shows the two cells after cytokinesis. So A is metaphase, and that was right. In the space provided, draw cell A. Label your diagram to show visible features. Now, notice this is a biological drawing. You are drawing what you can see and not a diagram of metaphase from your textbook. You draw what you can see, and you draw it reasonably large. And there are rules for biological drawing. Number one, you use a pencil. Number two, your lines are continuous and there is no crossing of lines. You don't do shaded lines, sketched lines. They're just one continuous line. All of your lines join. You label using lines and not arrows. And you should put in a title and a magnification. So those are all the things that should be in your biological drawing. So here is me making an attempt at drawing cell A. Notice I have drawn what I can see. It was a bit of a pain to do, I'm going to be really honest with you. And it is far from perfect. And label your drawing to show visible features. So I've labelled the chromosome, the cytoplasm, and I've kind of assumed that the outside of each diagram is the cell surface membrane. So here's the mark scheme. In order to get mark point 1, you had to draw one cell. It had to take up a reasonable proportion of the box. There had to be some attempt to draw the chromosomes. In other words, you weren't just drawing a diagram from your textbook. And it needed to be broadly circular. I mean, basically, you needed to make a good attempt to draw what you saw. So I got mark point 1. Clear continuous lines on the chromosomes and the membrane. Now, I'm going to be really picky with myself. This isn't great because I haven't, it hasn't got a nice continuous line. And places like here and here, where else? I mean, you can see where it's not perfect, where basically I haven't got the lines nicely joining up at the ends. There are no, I shouldn't have any overhangs, as it were. So I'm not going to give myself mark point 2. Ruled label lines touching the correct feature, yes, I've done that. And the chromosomes and the cytoplasm, yes, I've done that. Colour, I didn't mention that. So I've given myself mark points 1, 3, and 4. So I got 3 out of the 4 marks. The volume of cell A is that number there. Assume that it's spherical. Here we're given the volume, the formula for the volume of a sphere. Calculate the volume of cell B. Give your answer in standard form. Okay, all right. So I'm going to show you what I thought of doing first. I'm no massive mathematician. So I kind of recognized that this equaled this formula. And therefore, I could rearrange the formula to get R cubed, and therefore I could cube root that to get R. And then I kind of could look at the, I looked at the insert, and I basically kind of went on my picture, and now it doesn't matter what size of picture I was looking at, but this was kind of 10 millimeters from here to here, while this was 14 millimeters. So does it make sense that the diameter and therefore the radius of B would be 10/14 of the radius of A? And therefore I can now work out the radius of B. And now I can scale it back up, as it were, to work out the volume of B as that. Do you do the same? Oh, Ariba, do I do Edexcel? I haven't done an Edexcel paper. I could do an Edexcel paper. Let me bear that in mind. Thank you for the question. I will absolutely bear that in mind, because I am now thinking that I am, for the next few live streams, I am going to concentrate on going through past papers. So thank you for that. Here is the mark scheme. Now, I was working back from information given about cell A, and because I got 1.2 times 10 to the 4, I got 2.2. I was within the error that they allowed, plus or minus 0.4. That basically says that it would go down as low as 1.8, and they could accept anything up above to up to 2.6. So I was absolutely fine. But let me show you the simpler way of doing it. So I got the marks, fine. But now have a look here. If I had just, again, using my measurements, where I had measured the diameter of A as 14 millimeters and the diameter of B as 5 millimeters, notice that that makes A about 7 millimeters, the radius about 7, and the radius of B about 5. Now, the only thing that changes in this formula is R, sorry, this should say R cubed. Sorry, I just realized that was wrong. The only thing that changes is the value of R cubed. So if I work out the relative fraction of R cubed of each of these, does it make sense that volume B, the volume of B, ends up being 1/25th of 343 over that fraction of volume A? And therefore, if I had gone down this route, I would have actually ended up with exactly the same answer, but quite possibly more quickly. All right, so just showing you that bit of maths as well. Thank you, Ariba, for the tip. Thank you. State the type of microscope that was used to view these images. Well, I'm looking at that and going, it's absolutely a light microscope. I can only see chromosomes. I'm not seeing any other organelles. And it's in color. You only get colored images from a light microscope because the light microscope uses white light, and therefore you can see color. Electron microscopes use electrons. You cannot see color with electrons. So light microscope because colored stains were used, kind of red, and only chromosomes visible, not organelles. So here was where I got the mark. I didn't talk about the absolute magnification possible within a light microscope. I'm afraid that information isn't right at the top of my head, but the color absolutely is, and the fact that there was no organelles was the other thing. This is obviously valid as well. Mitosis involved in growth and repair. State two other roles of mitosis in multicellular organisms. Well, easily I thought of asexual reproduction. So multicellular organisms can reproduce by asexual reproduction. You have plants reproducing by rhizomes or what else are they? Or bulbs or runners or suckers, all of those kinds of things, all of those extra stems that they produce. And I was really struggling to think of something else that wasn't asexual reproduction, because I kind of growth and repair of tissues. I thought about the cloning of T lymphocytes, but I wasn't convinced. So because I just kind of thought, oh, well, that's just cloning. So I didn't write another answer. Here's the mark scheme. Asexual, absolutely, I got that one. So I got mark 0.1. Didn't think of any of these and didn't recognize them quickly enough as being an alternative to the type of asexual reproduction that I was thinking of. I was thinking of the asexual reproduction of the organism, but in my mind, I was just kind of going, well, mitosis is asexual reproduction, so wouldn't that be included in the cloning of B lymphocytes? So I didn't think that one through clearly enough. So, anyway, it was only one mark, so I got the flipping. Oh, did I have to get? I didn't get the mark. I've just realised I wouldn't have got that mark because I didn't put 2 down. So anyway. Okay, on to penguins. This is in Antarctica. This is showing how the population of the Adelie penguins reduced. This is showing, this line is how the chinstrap penguins increased. And this is the Gen 2 penguins and how they were introduced and increased from about 1993 or something like that. So before 1975, the only penguin was the Adelie. Chinstrap penguins were first recorded on the island in 1976. Okay. The changes in the chinstrap penguin population are not directly related to abiotic factors. Right. So they're not directly related to things like temperature, availability of water, what else? Wind, all of those abiotic factors. I'm not really thinking of soil conditions because it's Antarctica, it's covered in ice. Suggest explanations for the changes in the population of chinstrap penguins between 76 and 2010. So this is what I wrote. Chinstrap population increased because it outcompeted the Adelie penguins for resources such as food, because I'm noticing that as these increase, the Adelie penguins are decreasing. The chinstrap population plateaued because of the increasing Gentoo population competition from the Gentoo population, which outcompeted the chinstrap population. Do you know, actually, thinking about that answer, it didn't outcompete the chinstrap because the chinstrap penguins didn't disappear. So actually what I've written here is not correct. What I should have said is that the chinstrap penguin population plateaued, and that was because of the increased population of the Gentoo penguins. So here is the mark scheme, and these are the marks that I gave myself. Now, I should not, I've just realized I should not have given myself I don't think I should have given myself mark 0.5. I talked about competition. One of those two marks. I'm struggling to work it out immediately now. Which one? One of those two I wouldn't have got. So I would have only got 2 out of 3. Calculate, and just looking at the examiner's report, 1 and 2 were hardly ever given, interestingly, and I must admit I didn't think of this thing. I probably should have thought of mark point 4, but I didn't. So, you know, hey ho. Calculate the mean annual decrease in the mean annual decrease in Adelie penguin population between 88 and 2010. So I have not done this brilliantly, but I've tried. So I've taken a line up to 88 for the Adelie penguins, and I'm thinking that that is about 1,180-ish. Not entirely sure. I couldn't put these lines in exactly the right place, but I'm thinking that's probably about 220. So this is what I wrote. 1,180 minus 220, and then I looked at the y-axis again, at the label, and it says that these numbers are divided by 10 for the Adelie penguin. I've got to times my answer by 10. And therefore, that equals 9,600. But that decrease has occurred over 22 years. So I'm then down to 436. But then I read again and kind of go, oh, hold on. Those are the pairs of penguins. So I've now got to times this number by 2 to get the number of individuals that died on average each year over 22 years. And it was 872. Now, interestingly, I would have got the marks for either working it out for the individuals or just for the pairs, because this should have actually said pairs per year. And I was within the within the bounds of accuracy. So this number plus or minus 40 got you the 2 marks. This number plus or minus 20. Okay. Right. Adelie penguins need a habitat that contains sea ice. Gentoo and chinstrap penguins can survive without access to sea ice. Scientists have claimed that the population changes in 3 penguin species on the island suggest that the Antarctic temperature is increasing. Discuss whether the information supports the scientists' claim that the Antarctic temperature is increasing. Excuse me, you should refer to data. Okay, well, given that the Adelie penguins rely on sea ice and their population has decreased, and the other 2 penguins don't rely on sea ice and their populations have increased, that is an argument for, or that's an argument supporting their claim. Notice that this is an evaluation question. You are basically kind of going pros and cons, for and against, supports and doesn't support. So for, I kind of wrote, and I wrote what I've just said to you. And then I realized, refer to data. So I thought, okay, now I've got to talk about what happened to the Adelie penguin numbers. So that's what I wrote. The against was that the population changes could be due to some completely different factor. And rather than just saying, could be another factor, try and think of something. So I kind of thought of something that could cause the Adelie penguins to reduce, and that would be a new predator of some kind. And I then went on to say that correlation with temperature changes does not equal causation by temperature changes. So that's where I put. So I got these mark points. I actually got 4 marks for my answer. Obviously, I would have only got 3. They monitored water temperatures and populations of other water animals. Okay, suggest 2 further pieces of evidence that the scientists might have found to support their claim. Well, if they monitored water temperatures, they would have seen, obviously, that the ice cover reduced. So I put reduction in ice. I couldn't actually think of anything else. Here's your mark scheme. Look at all the things I could have written. I'm pleased I got mark point 1. Do you know, I'm going to be honest. Do you know the one that I think I should have got is mark point 7, but I didn't think of it, so I didn't write it, and I only got 1 mark. Right, for centuries, artificial selection has been used to improve the quality of crop plants used for human consumption. Explain with reference to selective breeding why it is important to maintain viable wild populations of crop plant species. So my immediate thought is that the crop plants that we have come about by stupid amounts of selective breeding. That means we have massively reduced the allelic variety in those populations. We have nothing like the number of alleles in the population of our current crop plants because we have selectively bred out most of them. So why do we want wild populations? Well, my thought is that if something happens in the future, like global warming, climate change, or a new disease for one of our current crops that we're dependent upon, we would be reliant, we would be desperate to then breed together those wild populations to bring about a new strain of crop plant that we could use under the those current conditions. So this is what I wrote. Which is pretty much what I have just said. Selective breeding reduces the allelic variety of climate change or new crop disease, wipes out a particular crop plant, then the wild crop plants can be bred to produce a replacement strain. And here is, now it's got an asterisk, so it is one of those leveling quality of written communication questions. The science determines the level, your degree of relevant and accurate communication determines the mark within the level. So reading through my mark and reading through the scientific points that could have been included, I think that my answer is probably either a 4, because there isn't any irrelevant information, and it all uses appropriate scientific terminology. There's a reasonable amount of reasoning and structure. I think it's either a 4 or it could be a 6. Now, I'm going to give myself a 4 because I'm not going to overmark myself. So I'm going to say that, because I think there's a possibility that I could have included some more scientific information. So, question 19. 2 students investigated the growth of bacteria at different temperatures. So they had flasks of nutrient broth. Flask 1 was inoculated 1 centimetre of broth containing the bacterium Bacillus subtilis and incubated at 20 degrees C. Flask 2, same again, but at 30 degrees C. Flask 3 was inoculated with no bacteria and incubated at the higher temperature. Notice the temperature that's more likely to bring about rapid growth. Aseptic techniques were used throughout. At set times over the next 3 days, the students removed samples from each flask and measured the numbers of viable bacteria present. State one further variable that they should have controlled in order to produce valid results. In other words, you are investigating what you think you're investigating. And the obvious time to me is that these samples need to be of a controlled volume. You've got to have, those volumes have got to be the same. And that was mark 0.5. So I got that mark. And then it goes on to describe, hold on, that's not in here, that's the next one. And then we go on to describe how the original flask, the contents of the original flask were diluted, and then each of those dilutions, 0.1, was put onto an agar plate and cultured. So I've drawn a little diagram of what went on. We had our original flask, 0.1 centimetres cubed, was put into 9.9 of water. That is an 100th dilution, and that was A. Then 1 centimetre of that was put into 9 centimetres cubed of water, or sorry, not of water, of broth, and that was B, 10th dilution. This was repeated down to F, and then 0.1 centimetres cubed of the dilution F was put onto F's plate. And you are told to use tube F to calculate the number of viable bacteria. Well, there are 6 bacteria on the plate. And this 0.1 sample has come from, notice how many factors of dilution there've been. 1, 2, 3, 4, 5, 6, 7, because 100 is 10 squared, 10 times 10. So the answer is 6 times 10 to the factor 7. And the reason why it's 6 times 10 to the factor 7 is that 0.1 sample have been used here, and 0.1 of the sample is what was taken out. So it is just 6 times 10 to the 7. And that gave me the 2 marks. You got 1 mark for being out by a magnitude of either 10 or 100. So if you've got your dilutions factors wrong, you got at least 1 mark. Okay, the students disagreed about which tube's result to use as a starting point for their calculation. In other words, which was the better one to use. Discuss whether a petri dish resulting from whether tube F was the most appropriate one for them to use. Well, I would argue that tube E would be the most appropriate one to use, because actually there are more countable colonies on tube E. And you should be recognizing that each colony has come from a single bacterium. And therefore, you know, if you dilute down your broth enough, you end up being able to count the individual bacterium that were in that sample. And of course, they then divide each one to grow into a colony that you can then visibly see and therefore count. And my logic was that if there was either a counting error, or if you do replicates and you get slightly different numbers, your error was going to be less if the numbers were different here than if they were different here, which is kind of what I've written. So this is where, but oh, notice this last sentence, I realised I've really argued for E, but this is almost an evaluation question. Discuss whether it was most appropriate. They're looking for pros and cons. So at the end of the question, I was kind of going, what can I say that's for F being used? And it is still usable because there are few enough colonies to count. It wasn't like any of these where you couldn't realistically count the number of colonies. So I got 4 marks, so I got the maximum number of marks. Now, this was where I was running out of time. I haven't done this answer. It will be in the next catch-up live stream. But let me just finish off this question. So let me just finish off this question. Oh, I think hopefully that's still working. Explain the purpose of flask C. Well, that was to act as a control. And it was fundamentally to show that there wasn't any contamination. But I was basically saying that there weren't any extra bacteria growing under those experimental conditions. So here we go. Notice that it wasn't a control variable or a control group. It was a control, and I hope that is helpful. Suggest why the teacher told them not to grow the bacteria at 35 degrees C. Well, 35 degrees C, you are basically using a temperature that selects for bacteria that could potentially be human pathogens, because 35 degrees C is very close to our body temperature of 37 degrees C. And you do not want to be producing bacteria that could be human pathogens. So that's why all bacteria in schools are incubated at 20 degrees C, not 35. And finally, why would they carry out the investigation several times at each temperature? Well, that would be to increase the repeatability of their data. And it's so that they could calculate an accurate mean and calculate a standard deviation. And, well, if they did it, well, more than 3 replicates, they could carry out statistical tests. So I got the marks here. So I got 3 out of 3 there. Now, I'm going to finish now because I didn't have time to carry on with the rest of the paper. As I've said, I will post another video which completes this paper. But until then, I hope this has been useful. And this will now go up on the channel and you'll be able to watch it at your leisure. So go away and have a well-deserved cup of tea and a break, and I will complete this video over the next day or 2. Okay, see you soon. See you next Tuesday when I seriously hope to be going through another paper. And I think it's going to be AQA paper 3. And then I will go through the OCR paper 3 paper the following week. All right. And I will see you then. Bye bye.